Good morning, student. In last period, we learned the comparison of plant cell and animal cell. Now, we know that the cell is basic, structural, functional, and smallest unit of all living organisms. Cells are generally microscopic, therefore it cannot be seen with our naked eye. So, to observe the cell, we require a device called microscope. Microscopes are of two types, a simple microscope and the other is compound microscope. So, with the help of this device, we can see the cell. If we observe the cell under the compound microscope, the magnification of the cell is much more larger than that of the simple microscope. In the compound microscope, you see the compound microscope in my hand. This is a compound microscope. This is the base of the microscope, and this is reflector which reflect the light from the source and divert it into this base. If we place a glass slide on this base, then the light is also falls on the slide, and this is the holder of the slide. Now this is the lens which called objective lens because it near to the object, and to observe the object under the microscope we observe from this side. So this is the eye lens or eyepiece. To adjust for the clear vision into the slide, there are two screws. This is a major screw and this is micro screw. So this is the main component of compound microscope. Now if we want to observe the plant cell or onion cell under this compound microscope, then the aim is to observe the basic component of the cells in the onion cell, that is plant cell. So, what is the procedure for the observation of plant cell or onion cell under the microscope? So first we require a onion. We remove the pink covering of the onion bulb by using a forceps. Forceps is a type of a chipia or chimta using forceps or with the help of your hand. So if I remove in the shortcut, then I remove the peel by hand. Separate the thin layer of the onion peel from inside the onion ball. I separate a thin layer. If you see, this is a thin layer. This is a thin layer. Separate a thin layer which is in my hand, if you can see it. This is the thin layer. So separate a thin layer of onion peel from inside the onion bulb using a forceps. Cut the separated onion peel into a smaller piece with the help of forceps. put a drop of water put on the glass slide, and then we put this thin layer of onion peel on the slide. Now put a drop of water on the glass slide and place the onion peel. Put a drop of methylene blue. Methylene blue is a stain. This is methylene blue. In this bottle, methylene blue are there. So this methylene blue, if you observe, the lid of the bottle is attached with a drop of methylene blue. This is methylene blue. Now put one drop of methylene blue and a drop of water on the glass slide and cover it with a cover slip. In my hand, this is cover slip. Cover slip is either a square shape or a round shape made up of a thin transparent glass. We put a cover slip on a glass side in such a way no air bubbles will remain in the glass side, and simply press it so air bubbles are coming out from the cover slip. After covering the cover slip, the slide should be observed under the microscope. Now we place this slide at the base of the microscope in this way and observe from here. Now what are the observations from this activity? So the cell can be seen laying side by side, bound by the cell membrane and an additional layer called a cell wall. So the sides are laying side by side, bound by the cell membrane and an additional layer called cell wall is there, and a spherical shaped nucleus is present in the center of each cell. This is a nucleus. A nucleus, these are the nucleus of each cell. And this is the cell membrane. Cell membrane is surrounded by the outer covering called a cell wall. This is the cell wall. Cell wall is present only in the plant cell, and it gives its rigidity and definite shape. And at the center of the cell, you see a spherical round shape nucleus is present in each cell at the center of the cell. And you also see that additional outer cover are also present in the onion cell. Onion is a plant, and the cells of the onions contains cell wall as an outer covering around the cell. So what are the conclusions? Basic components of cell are cell membrane, nucleus and cytoplasm are there in the onion cell. These are the basic components of cell. In addition to this, one more component, there is cell wall is an additional layer present in the plant cell. The diagram is given here. This is cell membrane, nucleus, cytoplasm and cell wall. In similar way, we observe with the help of microscope animal cell, that is human tick cell. If we prepare the slide of human tick cell, we require as a stand methylene blue or iodine solution. In the bottle, iodine solution are there. And similar proceed. We require a small toothpick or matchstick and clean toothpick are insert our mouth towards the tip of our mouth and scrape gently. After scraping sometimes, this material is transferred on the glass slide. Now we put a drop of water on it and as a stand 1 drop of methylene blue or 1 drop of iodine is also put on the slide. After covering the slide, we observe the slide under the microscope and observations are there. Such type of tick cell are we seen in the microscope. In this cell, we see that nucleus are present, cytoplasm are also present. Nuclear membrane surrounded, nucleus is also present and cell membrane is present. But cell wall is absent in the cell, this cell, tick cell. Tick cell is a human cell, human is an animal and in animal cell wall is absent. So this is the basic difference between plant cell and animal cell. In plant cell wall has an outer covering is present in the cell. But in animal cell wall is absolutely absent. So today we learn to observe the plant cell and animal cell with the help of onion cell and tick cell. In such type of teaching, there is some limitation. We cannot see you the onion cell as well as tick cell physically. If you arrange at your village or town or city or your previous school and request your previous teacher, if he or she arrange to help to observe such onion cell and tick cell with the help of microscope, then you can understand very well. Thank you.